Welcome back to Dr. Liu's VCE Biology. Today's topic is the relationship between nucleic acids and proteins, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Given that molecule S is found at the core of this single-stranded RNA, ss-RNA, virus, it is concluded that S is the single-stranded RNA genome. The structure of this RNA is defined by its characteristic phosphate-sugar-base backbone. Both exons and introns are DNA sequences found within the cell nucleus. Since they are part of the genome, neither can leave the nucleus. However, the pre-mRNA transcript containing both is spliced to remove the introns before being exported. Therefore, only the exons are retained in the mature messenger RNA, mRNA, and are translated into protein, while the introns are degraded. For a secretory protein, the process begins with the synthesis of pre-mRNA in the nucleus. After maturation, the resulting mRNA is translated at ribosomes located on the rough endoplasmic reticulum, RER. The newly formed polypeptides are then folded and modified, where they may assemble into their quaternary structure, if applicable. Finally, the fully processed proteins are packaged into secretory vesicles and exported from the cell. Tryptophan, TRP, an amino acid, is an important part of cellular functions. Escherichia coli is a prokaryote naturally found within the human digestive system. The environment of E. coli partly depends on the food intake of its human host. When there are sufficient levels of TRP in the environment, E. coli can produce their own. This production is regulated by an operon. In an environment with low tryptophan, TRP, levels, the TRP repressor protein is inactive and does not bind to the operator. Conversely, when TRP levels are high, two TRP molecules act as a co-repressor, binding to the repressor. This binding changes the repressor's shape, allowing it to bind to the operator and physically block RNA polymerase from initiating transcription at the promoter. The mRNA template directs the production of enzymes that create tryptophan. This amino acid is a fundamental protein building block and plays a vital role in preserving the protein's proper spatial conformation. This section, weighted at 10 marks, 8.3% of the total 120 marks, assesses understanding of molecular genetics, focusing on two main areas: single-stranded RNA, ss-RNA, structure, and the tryptophan operon. Multiple choice question 3. The fundamental phosphate-sugar-base structure of the genetic molecule in ss-RNA viruses. Multiple choice question 4. The similarities and differences between exons and introns. Multiple choice question 5. The steps of protein synthesis and the secretory pathway. Short answer question 1. The structure and function of the TRP operon. 1a. The mechanism of TRP repressor protein activity in both low and high TRP environments. 1b. The respective roles of mRNA and the TRP amino acid itself. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.